Our next speaker is Sandra Siegert from Austria, who's going to talk about how to get from microglial shape to their function.
Okay, thank you very much and giving me the opportunity to present some of our work which we did the last couple of years in my group, which is related to microglia and the shape. So if you think in biology about shape, we know that in a very simple term, when we look at birds, they have very defined beaks, and we know that each of the beaks' shape has a certain feature if for example, the bird eats a seed, an insect, or plants. So here, clearly, biology determines that the shape are linked to a functionality. And a similar principle is existing for the nervous system in overall. When we look at the individual cell types here in just 3 brain regions as an example, you see the diversity of for example, the dendritic processes here, the cerebellum, indicating that there must be like differences how they process neuronal information. And talking about the nervous system, we know that microglia are embedded throughout this entire network and respond to the local environment. So when I was, and we also know that microglia, which we have heard already, have a lot of diverse function in which they're acting and responding to the response. And when I started to work on microglia in 2015. Before I was was only working on neurons, so I just stepped into microglia field and was very curious how we could terminology define the microglia. And initially, I was a little bit overwhelmed. Starting in the literature, there was like M2, M1, pro-inflammatory, anti-inflammatory, BAM, TAM. We come up, so we have a lot of like taxonomy issues, I had the feeling, because neither the morphology, nor the function, nor the gene transcription, we have a coherent signature. And since I basically concluded that taxonomy is a little bit a mess, which makes it difficult to have a common language and to talk about the same principles, what we are talking about. So initially, driven by the question how to get from microglia shape to function, I was like, let's first start with the morphology. Maybe it's not so bad as my first impression is. And my student Gloria started to look into these principles. She collected over like 10,000 adult microglia shapes across 7 different brain regions in biological triplicates for male and females. Involving the olfactory bulb, frontal cortex, somatosensory cortex, dentate gyrus, cerebellum, cochlear nucleus, and substantia nigra. And as we would normally do when we do the morphology, we go we generate a 3D skeleton, and then we have here a list of like different parameters which we can choose. And one of the parameters which seemed very appealing is, for example, the terminal endpoints, where we generally assume when we are looking in the microglia context, now I have here shown a frontal cortex, we would assume we count the number of terminal endpoints. Here in this example, we have health 16, and in the disease one, we have 9. So less terminal endpoints, the microglia is more reactive. That's the basic conclusion, what we normally do, or my impression was in the field. And so now I'm looking in the adult brain. When looking at these parameters, it becomes very difficult to distinguish the different morphologies. And we know that the frontal cortex and the dentate gyrus or olfactory bulb have different neuronal function. And so the terminal endpoint doesn't seem to be a good marker to distinguish. The only thing what we can distinguish on the statistical basis is maybe the cerebellum and the cochlear nucleus to the other brain regions. At this point, we were like, maybe we just have decided the wrong parameter. Another famous parameter everybody uses is Scholl analysis, in which you form concentric circles around the microglia and count how often the processes go through. We did this. Similar problem we were running to in the adult microglia context. We could distinguish the cochlear nucleus cerebellum, but in the other brain regions not. And furthermore, we have a huge error bar. This is standard error, and it's not standard deviation. So it's like really bothered me in a way. And then also it bothered me that we don't know which radius to choose. We can find in the literature everything from 1 micron to 10 micron, and each of these parameters has an influence on this. At that point, we were like, we have to step back. We cannot yet answer the question how to get from microglia shape to function, and we should rather think about how can we reliable and in an unbiased way compare microglia shapes across conditions, starting at the adult. And that was the time where my postdoc Ryan joined the project, and where we have been working together with our collaborators at the EPFL. And KTH, two mathematical teams, to identify a strategy which we then called morphomics. So let me point out what is the challenge we are running through if we analyze microglia morphologies. We normally generate 3D rooted microglia trees, like this is a branched structure, but they are very difficult to handle. We want to simplify the tree, and the trick is to simplify this complex 3D morphology in a way that you keep as many features as possible of the tree structure. So if you think about before, we had like the terminal endpoint, but then, the terminal endpoint. But if you only have the terminal endpoint, you might missing the branches or the branching length or the length of the terminal of the branches, which could be important parameters to distinguish the microglia morphology. To circumvent this, the mathematician had developed a strategy based on persistent homology, in which they reduced the tree complexity and generating something that is called a persistent barcode. And don't be scared, it's not like too crazy. You just generate a filtered function, and then the filter function going towards the root, so the cell soma, and it marks here when a process is born. So you will see it here as a start of a bar, and then it goes further and sees another bar start of the process, and will start another bar here. And then at the bifurcation point, it will make a decision. And decision will be the one which was born first will die. So that means this orange one will will stop to grow, and the blue one will continue. And that is going on. In that way, we reducing the branching complexity into bars. So we represent this 3D skeleton in a two-dimensional space in bars. And those bars we can then further collapse into like coordinates. So if you have thinking about this as like a B and a D, we can put it in a persistence diagram in a coordinate, and then we can use Gaussian distribution to generate a persistent image, which represents similar what we have seen in the 3D skeleton, where we have the soma here, and along the axis, the number of branching properties and how many branches are there reflected in the pixel intensity. And this allows us now to be more precise in the analysis. We did this analysis, it helped, but there was another challenge we are running too, and we're looking at microglia. And you see it very nicely, the microglia are moving. And each microglia moving and surveilling causes variability in the data set, because now we only have taken snapshots. And so we had to identify a strategy and statistical approach to deal with this variability which existing intrinsically. And to this, we're using a bootstrap strategy, which is like we have the microglia pool here, we take a subpopulation of the pool, we do the analysis, we generate an average persistence image, and put it into a bootstrap microglia pool. And we do this several times. We have controlled for this, for how often we iterate through. And in this way, we reduce the dispersion of the variability within the sample. And then finally, to represent the data in a way that is intuitive to understand, we used data visualization strategies, which we have heard today already about UMAP presentation. This whole structure we call morphomics, and with the three parts. And what we did now is we added those 10,000 adult microglia into this pipeline and to see how the spatial heterogeneity looked like of the microglia, and suddenly we could see it. So by what we could detect is indeed the cerebellum and the cochlea nucleus are still separated. At the same time, we see now the somatosensory cortex is separated, then the gyrus, the substantia nigra, the frontal cortex and olfactory bulb seems to be intermingled. I later come to it. There is a sex-specific signature here, which apparently is making this intermingling there. So overall, what you can see here, that it is possible to separate the microglia morphology in the adult healthy brain. It's just a way how to identify what are the critical markers to do it. Now, of course, you could say, this is a nice strategy. Maybe TMD is actually not the important one, and we would like to prefer to use our Scholl analysis because we are familiar. So let's look the Scholl analysis and just apply the statistical approach and the data visualization on it. So we did this control experiment, and we have... And what we see is that the Scholl analysis, still with a radius of 1 micrometer, is not able to resolve this complexity as we have seen with TMD. So it's truly the combination of combining the TMD with the statistical approach, which we apply morphomics to separate the morphological features. Encouraged by these results, we started to build a reference atlas. And we have now these microglia morphologies. And we first were thinking, is there sex-specific difference existing between male and female? This is an ongoing debate. We know during the development there seems to be a difference. In adulthood, you can find paper in the one or the other direction. And so we started to look into this. And indeed, we find that certain brain region, there is a sex difference. We have here the males highlighted in the color, and on the right side, the female. And each time we have like the frame around it for the vice versa for the sex. You can see that the cochlea nucleus, the microglia are different. The cerebellum and shows sex difference, somatosensory cortex, and in the olfactory bulb. So the microglia have a signature that suggests sex-specific differences, which is less dominant in the frontal cortex or in the dentate gyrus. And interestingly, it's always an ongoing debate when you do experiments with like microglia related to sex differences. There's often then the reviewer asking, can you do overectomy and see what are the consequences of this? I hope with this data you should think about if this is a smart idea, because what we have been doing is we overectomized females from P20 onwards and looked at the microglia morphology in adulthood. And you see that the microglia morphology is completely different. It neither represents a female or a male microglia, and it seems to be a complete other morphological shape and state probably in there. And even interesting, you see there is an intermingling between the frontal cortex and the substantia nigra. So just a word of caution about the overectomy. Now, having said this about building the reference atlas, this was an adult, we have differentiated male and female. Now, we started to look a little bit further into the different state of activations that are existing in microglia morphological. When we look at, for example, postnatal development, so we used 3 time points, P7, P15, and P22, and 2 mouse models in which are associated with a neurodegenerative disease. We have a fast progressive degeneration mouse model, that's a CKP25, where we selected 3 time points. It is a very fast degeneration by removing the CKP20, activating it within like 6 weeks, you have an absolute degeneration of the hippocampus. And then we have the familiar form of Alzheimer's disease, which is called the 5X-FAD model, where we looked at the 3 months and 6 months. Again, male and female in at least triplicates. And so we have been generated this map of, here I give you only one map of example of the frontal cortex to see what can we learn from this. We have here the female microglia in the frontal cortex. We have here the control cluster. And now in the development, you see that the microglia are moving to P7, P15, and then P22, morphological, and then going back to the control. So it seems like at P7, they are closer to the control morphologically than in P22 than P15. And P15 is a time point where a lot of synapse remodeling is going on in the cortex. Now, when we look at the two Alzheimer models, we see they are, at the frontal cortex, they are not yet as strong in the 5xFID. But still, you can see they have like a morphological shape similar you would see in this like early developmental states. And interestingly, when we start to look at this fast degenerative model, you initially see after 1 week not so much changes going on, but then after 2 weeks, the microglia morphology suddenly becomes very reactive and turns into this arm, and we call it a disease arm almost. So a very massive change of the morphology. Now, how does it look like in male? We see here the difference. We have here the control. And similarly, as with the female, we see at P7, P22, they are like separated, but still, and P15 is in another dimension in this representation. And when we're looking at the 5xFID, they are activated, but it's not as dramatic as you would see with the CKP25, where you have, like, interestingly here in males, already after 1 week, the microglia morphology is very much moving, 2 weeks, and then suddenly there is a jump from 2 to 6 weeks. So I'm just giving you this one taste of an example of a reference map we have in the publication, also the other maps, so if you're interested into looking this. So how can we now use these reference maps? And this is like the idea what we are thinking of. And one strategy is like the drug screening approach. So we have been exploring, initially when I started my group, we were thinking which anesthesia to use. Very simple question, you write an animal protocol, isoflurane or ketamine. We were like, I don't know, both, we have to think about it, have to look at it. And for us it was really striking, this is an observation what we have made, and which I cannot talk too much today about it, but in 2 weeks I give the microglia talk here, so I will talk more about this interesting effect. Where we have been seeing here, and this is the somatosensory cortex, and we can label the perineural nets. This is a very defined extracellular matrix structure which shields neurons from inducing changes, neuronal activity changes. And what Alessandro did, he repeatedly gave ketamine anesthesia. He gave it 1 time, waited 3 days, gave it a second time, and waited. And what happened is that after 3 times, the perineural net was gone, which was pretty striking because the idea is that this is like, once it's formed, it's stable. It keeps memory in state, and it's not like, so you are kind of rejuvenile plasticity. And he shows in his study that the microglia are critical involved in this, because if he doesn't show, if he depletes the microglia, he doesn't see this effect of the PNN remodeling. So at this point, we were like then wondering if we can see this also on the morphological shape in the microglia and in our reference map in the somatosensory cortex. And this is like the adult control microglia, and then here we have all the different conditions which I introduced to you, the development, the different Alzheimer models. And this is now the PNN looks like when you zoom in, where you have seen this like mesh around the neurons. And after 1 time ketamine anesthesia, there is not such a strong effect. But then when you look at the 2 times ketamine, you see that the microglia morphology is adapting, and indeed the morphology of the ECM, the PNN, starts to be like disassembling, and after 3 times ketamine, you see that all the PNN is already gone. And the beautiful is now, we can also now map what would happen if we look at the recovery. Because general anesthesia has the principle that the system, the brain, should go back to its first functional state. So nothing should be changed. It should be just a temporary point. And after 3 times ketamine, we're looking 3 days after. It looked promising, the microglia going back to the control. However, after 1 week or 2 weeks, the microglia turned up in another state. So they have morphologically adapt to this changed environment. And it's not anymore like recovering to the healthy condition as we have been seeing. And we can now doing like this, using this persistent images as an example, where we can subtract the persistence images and see which morphological features of the dendritic tree are changing. And you can see that after the 2 weeks recovery, that's then in blue, you have much more processes closer to the soma compared to the control conditions. So indeed, the microglia morphology has been adapting on this point. And this is now also where we're starting to explore a little bit more. This is more a practical example with the drug screening, but we're also we are still interested in which features, morphological features are relevant. And which of the features of the 3D skeleton are informative, because do we really have to always trace a 3-dimensional the skeleton? And we started very preliminary experiments at this point in which we used the frontal cortex. That was the female, where I've been showing you that there is a jump going on in 1 week not, and then after 2 and 3 weeks it's jumping. And what we can now do with the persistence barcode is we can just select the bars which are starting at the root, so at the soma of the skeleton. And when we do this, which is basically like the primary branches, we would say, we're just focusing on the primary branches, how would then this distribution would look like? And if you only include these long bars, you see that this distribution falls apart. It's not anymore so sharp as we had it before. And similarly, we can only just focus on the small processes, so the small bars, which are in gray, in the opposite way. And even there, the distribution doesn't fall apart and doesn't reflect when we include all bars. Now we can say this is a female phenotype. Let's see if it's a similar phenotype in males, where we had this nice trajectory going on stepwise from 1, 2, and 6 weeks. If you do the long bars, you completely destroy. There's a very nice separated morphology adaptation. And all similar with the short bars, it's like a massive polarization to the activation. And I think we have now started to explore here, and we are just continue to explore further what are the morphological features. But it's pretty clear that we have to analyze all kind of different features together to get a whole complete view of the microglia morphology. And we are also now at this point starting to explore, getting a little bit more into the morphological features, in looking into a perspective of like the microglia in the retina, because the retina is a very great example. Because now my presentation, oops, the video is not. Let's see. Doesn't start. Let's see. Maybe it froze the video. Okay. Doesn't want it. Wanted to show you how beautiful it is. So now you just have to live with the vipertome section, with the side view, where you have the photoreceptors here, and the microglia are sitting as a single layer in the outer plexiform layer, and then here in the inner plexiform layer. So they're very compartmentalized in the retinal structure. And this allows us to like systematically going through and showing the pattern of going on. So, and indeed, we generated a map of the morphological spectrum of the microglia morphology. Just to give you a taste of it, it's from the OPL, where you can see the microglia trajectory from every day from P6 to adulthood, where the microglia trajectory is changing in this direction in a very reactive way, and then trajectory coming back. And this is different to the microglia which are sitting in the inner plexiform layer, which show a different trajectory, starting much more reactive, and then, but still ending up in a similar compartment in the adulthood. And then to sum up further, we are now looking also in features that are associated with the microglia. Additional features within the microglia, like the mitochondrial distribution, which we think is also a very important parameter to predict the microglia morphology and will be informative for further insights. So in summary, what I want to say is that we have generated a reference list over 40,000 microglia. You can find the 3D morphologies all uploaded in neuromorph.org. And we generated a morphomics algorithm to reduce the tree complexity and information to user selection biases. On this said, we think this is like the Paulicelli paper, which is already several times pictures shown. I think we have addressed a lot of those points that have been shown. We're talking about mouse, the sex, genetic background, postnatal, different spatial location, and even drugs, and show that there is indeed a morphological signature there. So, and with this, I would like to thank my team. It has been involved in this. This is only one part of the project I could have been shown here, and I thank our collaborator. And if you wonder where is IST Austria, it is close to Vienna. And that is here, our building, where we are. And we have a lot of possibilities there. We are always looking for faculties, position, have postdoc program and graduate school and internship program. So check out the webpage if you are interested to come to us. And thank you for your attention.
Thank you very much. So, do we have questions? Yeah, in the back.
Fantastic talk. Thank you so much. It's really interesting. You're putting all those different features together and then generating that sort of skeleton map. But I wonder, what do you think about potentially like I know maybe people don't look at this very much, but the width of the microglia arm, for example. Have you thought about incorporating that into your data set at any point, or do you think that's important?
Do you mean the width of...
yeah, like the width of the process, for example, and how that might change, because obviously it will get thinner as it goes, and do you think that's important when we're looking at microglial morphology?
So we started this project in 2016, and at that time Imaris didn't include this one. We did the tracing feature, and it was like not... I know now Imaris has been including this feature, so we have not included it. I know SOMA is always a question. Have you looked at the SOMA size? I personally have a hard time to explain how a difference between a SOMA size between 10 and 12 microns is explaining a difference. That's my personal taste. But what we also now thinking is, like, for example, the angle of like certain dendritic branches. That could be a feature that is also involved, that is not yet fully integrated in here. And as I said, the organelle distribution, which we think is a critical parameter. So all of the studies, all of the traced morphologies, we have also the CD68 as an endosomal lysosomal marker there. And we also have now several data sets with the mitochondrial distribution across different time points, and then to trace exactly which branch contains a mitochondria and how it distributes. I think that will help us to even further refine the map.
Great, thank you.
Okay, Angela.
I was just wondering if the perimeter of the cell membrane changes in different states, because it's a sensing cell. So, it's got to go somewhere when it's received a signal, and whether it just changes its shape, but does it actually have membrane metabolism as part of that?
And you're talking about the mitochondria itself?
No, the cell membrane. So lipid biology, really.
Okay.
Phospholipids.
Okay, lipid biology. I didn't think about this. It's an interesting... You mean like... yeah. So you have to synthesize more molecules that you see that it
You just squash it into a different shape.
It's an interesting approach because this is something we are now starting to think in another project in which we differentiate human iPS cells, like how the environment influences the microglia morphology. I cannot give you an answer, but we are happy to discuss it later.
Any more questions? Yeah, there.
Beautiful tool. So I could imagine that certain of the parameters that you measure are dependent, that they report on the same process. Have you done dimensionality reduction and check if that affects the states that you find?
So it is actually not that actually these 24 parameters, or like this list of parameters, they are often dependent on each other. So there are often common studies like determinant endpoints and branching complexity. They are interconnected. Here we are purely focusing on the 3D skeleton of the microglia. There is no overlay or something. It's only one parameter, which is like this branching complexity reduced on it. So we are excluding all this like biases that you are mentioning, like having different features selected that are interconnected with each other. So I know there is like several studies putting like 60 parameters together and try to and emphasizing this like differences. The differences between the microglia, but there is like the danger that you have like this overlap, and then you bias your data into one direction.
Yeah, one more question, and then I've got a question up the back.
Yeah, this is really interesting. So I wonder, this is a 2D cut. If you were to cut the mouse brain through a different angle, and
what do you mean with 2D cut? Because it is all three-dimensional reconstruction. So these are confocal images, 3D is C-stack, 3D images, and which we reconstruct.
But the sections are still cut in 2 dimensions, so the degree to which you can reconstruct all 3 dimensions is going to be limited, isn't it? Or
no, not at all. Because if you make it deep enough, like 200 microns or so, we usually make the section. We select the microglia where we have the full 3D dimensional structure. We exclude the ones which are border, which are only halfly branched and halfly accessible, and then we can generate this complete 3D skeleton.
okay, thank you.
Wait, one more. Wait, no, actually, I'm going to ask a question. You can wait. I just wanted to know, because different regions of the brain in an adult mouse, human or whatever, have different numbers of microglia. So, for example, the cerebellum has quite a low microglial density, and the hippocampus has, the most, essentially. How does that change all this? I mean, the density, have you kind of rigged it back to the density?
So overall, when we looked at male-female differences, the density was within the brain region similar. So there we didn't see any differences overall. What we have, you are right, there are different number of cells, and we are just exploring this, how this could be potentially influencing the data. We cannot exclude. Whereas of course, it's also the density, how thick are, how many dense layers of nuclei you have in there, and which ECM composition you have. There are a lot of factors that we could include into this measurement, and so this question can have not answered as straightforward as you're anticipating.
Okay, thank you very much.